Thank you very much, Dorina, for this introduction. And so I would like also just to thank the chair of this project and the promoter of this project, because without you, Yolanda, it would have been impossible just to meet all those persons. So it was really a very nice adventure from a scientific point of view, but also from a human point of view. And I think it's very important to go on in science. So thank you very much. So what I would like to do with you is just to summarize in a few slides, and I will try to stay on 15 minutes allocated for that, on the key discovery that have been performed in the field of the preclinical studies performed in the context of this MyNewGut project. And so if we have a look to the first objectives, and I will start, I would say, maybe with some of the conclusions. We can say that we focused namely on the wet bran as a source of nutrients to try to know how some components present in the wet bran may have positive or negative effect on health related to the gut microbiota. We also discovered, and you will see examples, of some enzymatic activities of the gut microbiome, of the gut microbes, that are very important for health maintenance. And so it also opens new doors, not only in the field of nutrition, but also in the field of drug development in the context of obesity. And lastly, we could also point out some new metabolites that are issued from the gut microbiota, and that can play also a beneficial role. And sometimes you will see that some concepts in the field of nutrition and microbiota may be unraveled, taking this into account. So, dietary fibers, I will start by this because it is part of my title. We all have dietary recommendation on the amount of dietary fiber we have to eat every day. Depending on the countries and so on, it is around 30 grams of dietary fibers. But only on this slide, I won't go on into detail on the content of these slides. You can see that the definition of dietary fibers is very complex. And so there are a lot of different molecules that we have to take into account as dietary fibers. And since we have dietary advices, we have to know what are the physiological and biological effects related to the fibers intake. And it is true that in most of the countries, for sure, we take into account some colonic function. It is mostly the transit time, but also the fermentation globally, as well as other metabolic features. But there are few data relating which type of fibers finally are able to interact with the gut microbiota and thereby have an effect on health. And it really fits for sure in the definition of prebiotics, and so it has been lastly defined by the ISAP, and in which you can see on this slide that among the dietary fibers, only some of them will be considered as prebiotics because the fermentability of the dietary fibers is really a criteria. So for this reason, so in the MyNewGut project, we decided in collaboration with the industry just to focus on some dietary fibers that are prone to be highly fermented in the gut, to try to know if we can have an effect on obesity related to some types of components. And so it is true that the wet is a very important source of dietary fibers, but also for sure of carbohydrates and proteins. I will come back to this. And so from the wet, the industry may also prepare some derivatives, and those derivatives will be called the wet bran extract during this presentation, so-called the WBE. So we first focus on this type of extract to try to know in animal studies what are the effects. The protocol of one of the study is this one, so it is a classical protocol in which you can see that in fact the arabinoxyline oligosaccharides have been given to mice, and so it's high-fat diet fed mice, and then there has been a follow-up taking into account for sure the gut microbiota. And so the effects were quite impressive, I would say, when we compare to other types of prebiotics that we have already tested before in our lab, but also in other labs. And as you can see here, so with the addition of this wet bran extract inside the diet of the animal, so you can see that we can have a very significant effect on the body weight. It doesn't change finally the food intake behavior, at least in the animals. And you can see that there is a consequence on the fat mass gain, including a decrease of all the white adipose tissue. So we tried to unravel by which mechanism it may occur, and it was not so easy because, to be honest, we don't have the key yet, but it is really impressive as data, but for sure we related this effect to the changes in the gut microbiota. So when we want to unravel how the dietary fibers interacted with the gut microbiota play a role on the host homeostasis, so we also have to refer to the fact that when the gut microbiota is changed, it changes for sure, as previously explained, the profile of metabolites that are prone to interact with the gut. And so in the gut there are namely endocrine cells called the L cells, and some of the metabolites issued from the gut microbes, namely the short-chain fatty acids but also some bile acids, may play a role on the production by those L cells of hormones that are prone to regulate appetite but also insulin sensitivity, insulin secretion and so on, like the GLP-1. So there is a link, classically in the literature, between short-chain fatty acids and endocrine function. When we had a look to the effect of the VBE extract, or the wet bran extract, on the gut microbiota, we could see that as for many other oligosaccharides, there is a signature linked to an increase of bifidobacteria that characterizes indeed the administration of these compounds. But you can see here some changes also of other types of bacteria that could be interesting to study as potential new targets, maybe as probiotics or whatever, in the field of obesity. When we measure the short-chain fatty acid content in the cecal content, we were disappointed. There are no changes. And it is not the first time we observe this, so it doesn't mean that they have not been produced, but maybe they have been absorbed. We don't know for sure because we didn't perform kinetics, but we couldn't relate these short-chain fatty acids with the endocrine function. But we had quite interesting data because, as I didn't mention to you, but when the L cells secrete the peptide, which is the GLP-1, glucagon-like peptide 1, the active form that we can see here can normally be destroyed and cleaved by an enzyme called the dipeptidyl peptidase 4, called the DPP4, and then it inactivates indeed the peptide. What we saw in our study on VBE extract is that you have a decrease as the high-fat diet increases the activity of this enzyme, meaning that it may contribute to the destruction of this interesting peptide. And you can see that with the VBE extract there is a tendency to decrease the activity in the portal serum, and we were really quite surprised to see that we also could measure a decrease of the activity in the cecal content, meaning in the gut microbiota itself. Then we asked the question, finally, could the gut microbes have a DPP4 activity? And indeed, in the collaboration with all the partners of this work package devoted to the animal studies, so we could see that indeed, so when we compare the DPP4 activity in the cecal content of germ-free mice who do not have any microbiota as compared to colonized mice, for sure you have a huge difference, meaning that indeed there is a DPP4-like activity in the gut microbiota. Then it has also, you can see that it is quite specific because we don't have such a change, such a difference between germ-free and conventionalized when we measure the DPP4 activity in the tissues of the host. So it means that DPP4 activity exists in the gut microbes. We know also that there are some drugs that can be prescribed to control diabetes and metabolic disorders, which are the DPP4 inhibitors to control the host DPP4. And then we just had the hypothesis that maybe those drugs may also modify and modulate the DPP4 activity of the microbes. And so it was the protocol here, and so we had mice with a controlled diet, Western diet once again, and Western diet with a DPP4 inhibitor, which is a drug called vildagliptin in the mice. And then what we could see that indeed the hypothesis was true. We can see for sure, due to the drug, a decrease of the serum activity of the DPP-4, but it was expected. But you can see here that also in the cecal content, in the animals receiving the vildagliptin, you completely blunt the DPP-4 activity, meaning that the drug maybe also acts on the microbial activity of DPP-4, which could contribute to health effect. And once again, concerning the health effect, we were quite surprised when we had a look to what happened in the intestine. These DPP-4 inhibitors are prone to act on the glucose homeostasis. What about the effect on the intestines? And to make a long story short, so we can say that when having a look to the ileum, we could see that indeed there were changes in the profile of proteins that are involved in key functions and maintenance of key functions of the intestine when they were treated with vildagliptin, such as antimicrobials, peptide and regulatory peptides like REC3 gamma, but also we saw an increase of some proteins that are prone to produce anti-inflammatory molecules and some proteins that are also involved in the control of inflammation and immunity. So it means that for the first time we show that these drugs, maybe by acting on the gut microbiota, may also improve the gut function. Concerning the change in the gut microbiota, it was also nice to see that when you give this type of drug, you change the composition of the drug microbiota. So we don't have a full view of all the changes performed, just to show you here that indeed we could see that some bacteria like Oxalibacter were decreased by the DPP-4. And so concerning these effects, so it is a direct effect of the DPP-4 inhibitor on the bacterial growth. Then it is one example of the context that when we have to treat patients, and obese patients often receive some drugs, the drugs may change the gut microbiota and some key intestinal function. We also saw some changes due to the vildagliptin to pro-inflammatory bacterial components that may explain maybe the gut effects. And so you can see here that the activation of some receptors by bacterial components present in the gut can be decreased by the DPP-4 inhibitors, and there is an increase of the propionic, which is a mediator of the inflammatory effect. Then DPP-4 is quite important. We can see in this type of assessment that the DPP-4 inhibitors may play a role on the gut microbiota and thereby have new effects. But we also know that the DPP-4 is also cleaving other types of peptides. Some cytokines, some peptides involved in the immune system are also cleaved by the DPP-4, as well as some molecules like gluten, which are the proteins. And then what we did is that we tested the hypothesis that maybe we could have an effect due to the administration of the prebiotics that modulate the DPP-4 to try to know if we can change the effect of gluten. And something which is well known with gluten, it is the gluten intolerance, but we see here by giving the gluten together with a high-fat diet in mice, as you will see after, the gluten may also promote obesity, and you will see the effect of prebiotics, including the wet bran extract we have tested, in this protocol. You can see here that indeed when you give the gluten in a high-fat diet, in fact, you worsen the occurrence of obesity, you worsen the increase of adipocyte size, and very interestingly, when you add the prebiotics inside the high-fat diet and gluten diet, you decreases all these effects. Then it means that yes, indeed, some prebiotics developed in this project are able to counteract the gluten and high-fat diet effect on obesity, maybe through an action on the digestion on gluten.
it was indeed quite interesting to see that when you give the prebiotics, you decrease the amount of gluten immunogenic peptide in the intestine. And so it's once again an interaction of some prebiotics, new interactions of prebiotics acting maybe to the activity of these enzymes, and this was correlated to the change of bacteria, namely with the Bifidobacteria and Prevotella species. Then I come back to my first slide by giving a more precise conclusions in view of the data I have shown to you, is that the red bran extracts are a source of nutrients, and so we have seen that VB extract is really a prebiotic fibers, changes the gut microbiota with some new effects related to those changes of the gut microbes. We could see that there are common enzymatic activities in the host and in the microbes, and the DPP4-like activities have been shown in this project, and this can have an impact on drug development and nutrition. And I will just show you, if I have 2 minutes, I have 2 or 3 minutes, yeah, to show you 2 or 3 slides just on the last topic, in which so we could also unravel new metabolites produced by the gut microbiota. So, as shown this morning so beautifully by the people from Iran, so the proteins are for sure very important to take into account when we have just to underline the role of the gut microbiota. And quite often we hear the people say, oh, but when the gut microbes take care of the proteins, so it's really harmful, it's not good for health. And it is true that we see more and more papers going out, and there will be some in the next week by very famous colleagues, showing that some metabolite issued from amino acids metabolism by the gut microbiota may play a harmful role in the context of obesity. But in our lab, so Martin Beaumont was able just to show that when you just have the metabolism by the gut microbiota of some specific amino acids, you may sometimes have also potentially beneficial effects. And so here are the amino acids we put forward in our project. And so what we did is just to try to analyze from these amino acids which metabolites can be produced, but it was already known. So, and for example, you can produce indole from tryptophan by the action of the gut microbiota. And these metabolites may enter the blood, the portal blood, and then reach the liver. And so we wondered if the presence of those co-metabolites produced by the gut microbiota from amino acid could influence the inflammation occurring in the tissue, and the inflammation driven by a bacterial component once again, which is the lipopolysaccharides. And we used the specific models of precision cut liver slices, which is a model quite interesting because it can be applied later on after this project on human samples, and it keeps alive, I would say, all the cell types inside the liver tissue, including the Kupffer cells, which are quite important when we talk about the inflammation in the liver tissue. And so to make a long story short, only one slide of data. So Martin Beaumont has analyzed in vitro but also in vivo experiment in mice the impact of those metabolites on lipopolysaccharides inflammation in the liver. And he could see, as shown on these figures issued from the in vivo data, that when you put you have LPS treated mice here, you have a huge increase of most pro-inflammatory cytokines, including the expression of genes leading to the NO and nitric oxide, but also oxygen-related species precursors. And when you have the indolic compounds present in the liver tissue, you can decrease significantly the pro-inflammatory process and the metabolic alterations. He could unravel the mechanism behind it, but the papers is now available, and so it is mostly due to changes in the NF-kappa-B pathway inside the liver tissue. Then it gives the example of one co-metabolite that can be protective in the context of LPS-driven inflammation, knowing that the low-tone inflammation occurring in obesity may participate to inflammation in the liver in the obese subject. For sure, it is my studies. Let's be cautious, go to the human studies, having now maybe new biomarkers to measure it in view of this project, but let's sort out this idea that amino acids are bad for health to the interaction with the gut microbiota. So here is the figures recapitulating this, but we have now several review papers published in the context of this MyNewGut project, and now we can put the indolic compounds also as actors of the modulation of the liver tissue in combination with all the very complex other mediators that can play a role on the liver tissue regulation. And so I would like to end here by thanking all the persons in the lab that have been involved in this project, and in particular the very talented postdoc that I had, so Martin Beaumont, Marta Olivares, who is here, but also Audrey Nering, who is really doing a lot of job in this context of this project and other projects in the lab also, and to thank all our support and the European community for the support in this project. Thank you very much for your attention.